So we're moving to the session called Breakthroughs in Foundational Research, and the talks we're going to hear have been previewed, I think, by the earlier speakers, and both of the speakers in this session have been referenced already. So we're joined by these two experts who are going to share the progress made in foundational research in forensic science in recent years. First, to discuss latent fingerprints, we welcome Dr. Joanne Buscaglia. She is a research chemist with the FBI laboratory in the Counterterrorism and Forensic Science Research Unit. And her research is primarily focused in the areas of microscopy, microanalysis, and elemental analysis of trace materials, impression and pattern evidence, and the interpretation of data in a forensic context. She's currently a Science and Technology Fellow of the Office of the Director of National Intelligence, a member of numerous boards and committees with expertise on latent fingerprints, and the recipient of the FBI Director's Award for Outstanding Scientific Achievement for the Black Box Latent Print Examiner Research, and the Paul R. Kirk Award, the highest honor given by the American Academy of Forensic Sciences Criminalistics section. So she will be followed by Robert M. Thompson, who's going to discuss tool marks and firearms. He's a senior forensic science research manager and the special programs office with the senior forensic science research manager with the special programs office of forensic sciences at NIST. And he has told me that to save time that I should tell you that his bio is in the program and move along so he can use that time for his presentation. So I advise you to look at your programs for more information about both of these distinguished speakers. They'll do their presentations, they'll give their presentations, and then after that we'll have some time for questions for both of them. I'd like to invite both of them to come up at this time and okay. Let me bring up your. And there's a clicker, or you can use the.
Okay, thank you. Thank you, and good morning. Can you all hear me? Yep, okay, I'm getting some nods. Well, I appreciate the lead up. I hope this presentation meets with your expectations. I was asked today to speak primarily about latent print foundational research that's been conducted since the National Academy's report. As Mr. Peter mentioned earlier, the FBI laboratory has been involved in research in many areas of forensic science for quite some time. And it's no surprise that prior to the NAS report, we had embarked on a research portfolio in latent prints that I will speak about. I will say, all of our disclaimers. I don't think I'm speaking about any commercial products, but if I am, it's for identification purposes only, no endorsement by the FBI. And these are our views, the authors and co-authors, my own as well, and not the official policy or position of the FBI or the US government. And then just some acknowledgments, just in case we don't get to them before the end, I'd like to do them up front. The work was funded by two divisions of the FBI, the laboratory and the CJIS division. And also a big thanks to the people who participated in these research studies. We could not have done them without the fingerprint donors, the latent print examiners, the laboratory systems that agreed to give people the time and the resources to complete this work. My colleagues and researchers, especially Brad Ullery, who sadly for us, but great for him, has retired this year. Austin Hicklin of Noblis, and Tony Roberts, who are here with us, as well as our more recent additions to the research group, Tom Busey, which will speak a little bit about the work in eye tracking later. Okay, so a little bit on the background. How did we get to where we are? Unfortunately for us, the impetus for much of this research portfolio was the Madrid fingerprint error, as it's commonly known. The FBI laboratory in 2004 misidentified a fingerprint to Mr. Brandon Mayfield as being involved in the Madrid train bombing. And as a result of that error, The FBI laboratory was very proactive in setting up reviews and committees to try to understand why that error occurred. And I was asked to serve on one of those committees. It's known as Team 8. And in 2006, we published the recommendations of that committee. My colleagues, Dr. Bruce Bajoli and Dr. Schwartz-Perlman, were also involved in that study. And you should be able to go get the results and recommendations online at Forensic Science Communications. And all of the references that I'll be speaking about today will be at the back of the presentation, so they should be easy for you to get them if you're interested. So as a result of this Team 8, we put together a portfolio of research that we published in 2006, but it actually started in 2004, 2005 timeframe. And so well before the NAS report came out, but it does align well with the sorts of things that the NAS was looking for. So we sort of had a head start. We began with evaluating the latent print examination process from start to finish. Much of the research that I'm going to be talking about today was in collaboration with Noblis. I will mention a few studies that were not done with Noblis or that were done before Noblis's involvement. As Mr. Peters mentioned earlier, we have published about 12 publications in scientific journals at this time, and there are more forthcoming. And we were very pleased to see that the PCAST report had a very favorable impression of the work we've done in the black box and white box studies, and we'll explain a little bit more about what those are in just a minute. Just as an overview of the research that was done, both back to the Mayfield Madrid misidentification up until today, and the PCAST report, essentially, we did look at the scientific basis, as I mentioned, that Team 8 report. There was the beginning of a persistence permanent study, which Mr. Peter mentioned. It started back in the early 2004 timeframe. And when we felt that this was something we should look at, we began a short-term study and then embarked on a longer-term study, which ultimately got published in 2019 by my colleague Keith Munson and his research group. We also had, as a result of the Madrid error, we had a Department of Justice Inspector General report, which also reviewed this portfolio of research and essentially agreed that we should continue with it. And so we looked at things like latent quality. What's the quality or clarity of the latent print? How do we assess that? The black box studies, which you've heard about. And then came the NAS report. So by the time that was published, we were well underway to writing and publishing the black box report. And then we followed on with the white box studies, exclusion studies, and eye tracking studies, which I'll talk about in just a moment. So what are black box and white box studies? Mr. Peters gave a quick overview, but we'll talk a little bit more in detail about them. In general, they're empirical tests of accuracy. How correct is the result, as well as the reliability, how consistent both inter and intra-examiner studies. They're limited to assessing the outputs of a process, really not looking at how that process occurred. And specific to forensic experts, the black box evaluations assess the examiner's conclusions without regard to how they make that conclusion. The white box analysis, the white box studies in general are also empirical tests, but they look at how a process is conducted. So they're not necessarily interested with the accuracy, although you may be able to design them to assess accuracy as well. But they're really designed to looking at how the process is conducted. And they're intended to model, define, or quantify how that process works. For forensic experts, the white box evaluations assess how the examiners conduct their examinations, as well as why they make the conclusions that they make. So why should we do these black box and white box studies? Well, black box studies are necessary to establish the validity of the forensic disciplines that rely on human decisions. Our earlier expert, Dr. Albright, mentioned the black box and white box studies. And one of the things he said was it was interesting to know about the process, and that's a white box study. The white box studies are necessary to collect detailed data to define these policies and practices, and as a basis for more objective or quantitative processes. Now, some of the disciplines who are interested in doing black box and white box studies are interested in jumping right to white box studies. Why do black box studies? Let's do it all in one study. I would caution against that, because one of the things that we learned from doing our black box study is really where to focus the resources required for a white box study. If the comparisons that we're using in our study are very easy comparisons, all examiners agree that it's an exclusion, or all examiners agree, or the vast majority of examiners agree that it's an identification, there's really limited value in testing those for the white box study. It's really not going to tell you anything about the process. So what we used the results of our black box study to help us refine and really zoom in on that area that needs to be looked at in the white box studies. So overall, let's look at the studies as we conducted them, and you'll see how they actually build on each other. We learn from one study and use it to answer a question or design the next study. So this is the latent fingerprint quality or clarity study. We wanted to know how can we standardize and quantify quality, clarity, and distortion of latent prints. And what you see here in the graphic is, on the top is a latent print, and on the bottom is a color-coded quality map that came out of this work, where the green is a good area, the red is a bad area, yellow is in between, and sort of a guide for where you should be looking and working. These guidelines and metrics and software tools were designed with the latent print examiner's involvement because we surveyed them, we gave them images, we had them use this color coding system and mark them up, and then use a training algorithm and some tweaking to get a software tool. The results of that were quite dramatic. We had an ANSI standard for specific markup and documentation of the friction ridge clarity. That's available at NIST ITL. The actual tool is incorporated into the latent quality metric software, which is part of the FBI Universal Latent Workstation, or ULW. It's been there since 2014, and it is widely used operationally. It's resulted in 3 publications as well, and we are happy to know that people are using this tool, and look forward to feedback and perhaps more refinement. The black box study was the next study that we worked on, and this actually involved a lot of upfront work that most people don't realize. You have to create the data sets. You have to create these where you know the ground truth. So you have to create these artificial samples that basically are very similar to casework. The black box study, as you've heard it discussed, was essentially the first large-scale study assessing the accuracy, reliability, and repeatability of the latent print examiner's decisions. In the graphics, you see a comparison pair. The top is the latent print. It's on galvanized metal, developed with cyanoacrylate fuming and gray powder. And lower image is the comparison image that it was being compared to. This actually resulted in one of the false identifications that we detected in the study. And this was a very, very difficult sample, very difficult substrate and development process. The key findings of this work were essentially, there was a very low erroneous identification error rate. The erroneous exclusion rate was considerably higher, anywhere from 5.5% to 7.5%, depending upon how you measure it. And then in the reproducibility and repeatability work, we saw 87% of the identification decisions were reproduced. So as a result of this study and its publication, we saw changes to policy, procedures, training, and quality assurance at many levels and many forensic science organizations. It's widely used in admissibility hearings. It was, quoted in the PCAST report as an example. And we had 2 publications, and again, these are at the back of the talk. So that was one data collection. We had a number of iterations where when we gave the repeatability and reproducibility samples back to the examiners, they didn't know that they were being tested for repeatability or reproducibility. They just thought they were doing extra samples that we needed. So that data collection finished. Now we do a new data collection with Whitebox, where we focus on collecting examiner markup and trying to get to the process of what the examiner is thinking, what they're doing, and why they help us realize why they make their decisions. And so the first thing we looked at was value decisions and assessing how image clarity and the quantity of features are associated with their suitability assessments for latent prints. The key findings here essentially were that minutia count explains a lot of the value decisions. Very little of those were affected by things like clarity or other features. The value decisions did vary significantly among examiners, and it's generally due to the difference in minutia counts. And we'll speak a little bit more about minutia counts in a minute. But examiner assessments were really not well described by binary choices. They had to make a value, no value decision. And when you're on that line, sometimes on the more difficult prints, the decision is not reproduced. Same data set, still black box data set, we looked at what was the sufficiency for individualization. So you have the suitability of the latent print, that's assessed first. And then you have, is it sufficient for an individualization? So when and why examiners individualize was our first question, and this is assessing both the quality and quantity of features that they use to make an ID rather than an inconclusive decision. And I'm not going to go into the graphic. Please see the paper for more detail. But for key findings, the significant variation in minutia markup was remarkable among examiners. The differences in corresponding minutia counts really understate the variability. So similar counts of minutia, if you're looking at different features, you actually could have very different minutia counts. So you really can't just rely looking at the differences between, let's say, 8 and 10 minutia. It's not the whole picture. The ID conclusions were driven by corresponding minutia counts, not clarity or other features, and many examiners seem to have this implicit decision threshold around 7 minutia for sufficiency, and this is possibly an artifact of previous standards where the people used 7 points either as an administrative threshold or a quantitative threshold, and then those people, after that threshold was gone, those people taught other people who taught other people, et cetera, and it just became ingrained in the discipline. Okay, again, same data set, different analysis. We looked at changes between analysis and comparison. How do examiners change the features that they marked after viewing the exemplar? So here I don't know how well that reproduced. Oh, that's not bad. It's not very good on this screen. So you see, right here you see on the top a latent print that's in analysis mode, what the points that the examiner marked in analysis. This is an extreme example of how the points changed when they went to a comparison, which is the lower level, the lower image. And this actually resulted in one erroneous individualization. I cannot show the comparison print because of IRB restrictions, but this was all done on the latent, both before and after, but in analysis versus comparison. So some of the key findings, every examiner revised at least some features during the comparison. The examiners almost always added or deleted minutia when they were making individualization decisions, and that was 90%. And as I said, the image on the screen shows you that this is an extreme example changes that one examiner made between analysis and comparison that resulted in an individualization error. Same data set again, looking at inter-examiner variation on minutia markup. How does minutia selection differ among examiners? And I will show you in this graphic, as it starts, what you're seeing are the markup of 15 different examiners who were asked to mark all the minutia that they see in this fingerprint. So it's, as it runs through, you'll see some are pretty consistent, others are different. But they were asked to mark all of the features, not just ones that they would rely on. So reproducibility, we found for minutia, is primarily driven by clarity, and it's also affected by the agreement among examiners on whether contiguous areas or region of interest, as well as value or comparison determinations. Overall, now we're going to conclude the data collection for the white box studies, and we have 5 publications that resulted from that. Moving on to latent print exclusion, we were asked as a result of the work we did in black box, one of our colleagues asked, have we looked at exclusions in more detail? So we went back and we looked at why we were, because we had such a high false exclusion rate is the reason we went back and looked at it and assessed the factors associated with examiners' exclusion determinations, and especially the erroneous exclusions. So here's some key findings. When you look at the graphic, that's an erroneous exclusion that was caused by an incorrect rotation. We found that most of the erroneous exclusions were generally caused by either misinterpreting the pattern class, which is essentially a level 1 detail, incorrect anchoring, or not finding valid corresponding area, and this we'll speak more about in eye tracking, misinterpretation of distortion, and excluding based on a single discrepancy. So this brought to mind that there is need for standardization. Generally, exclusion terminology varied among agencies, and there's no acceptable means to document the basis for exclusion. It's kind of easier to mark things that are in common, but when things are very different, how do you mark that? There's no really basic method for doing that, and that would may be helpful. This resulted in 1 publication, and we know of a few changes to policies and procedures, as well as training and quality assurance. As people become more aware that these exclusions, and of the exclusions and the high exclusion rate, the false exclusion rate, excuse me. Now we're on a new study, new data collection. We used some of the images from previous studies where we had a lot of examiners mark up in decisions, a lot of analysis on it, and we went and did eye gaze behavior. We looked at where the examiners were looking, eye tracking tools were used, and just a summary, you'll see key findings. I know I'm running a little long, and so I just want to move through. The graphic has a small box. In this particular part of the eye tracking, the examiners were asked to look at that area and find the target, kind of like Where's Waldo. We did find that when they were given the whole print with the box, which we consider to be visual context, it significantly affected their decisions as well as their rates of how quickly they could find the target. And once they were just given no context, which is essentially they had that box with the latent print detail or the fingerprint detail in it, but they didn't have the rest of the print around it, the lower image shows you where their eyes went. So kind of went to a couple of places in the print, not really zeroing in on the target as quickly as you see in the middle graphic. So this work was recently published online in a journal called Creepy, and we're hopeful for a second publication coming out next year. I am going to have to skip this due to time, but this is the work I mentioned earlier on the friction ridge detail. Mr. Peter mentioned it as well, and on the permanence, and an excellent paper from my colleagues, and go take a look at it. A little bit on implications, and I'm just going to try to wrap up quickly on this. The results of these studies really have had a variety of implications for the latent print examination process. We have seen some changes as a result of this, and the profession has improved. One of the nice things about this is that the profession has actually been very open and embraced the change, which is usually not what we expect. Nobody really likes change, but I think as we improve and move forward, there's been some good changes that have been made. The implications here for standardization processes and terminology should be obvious. The need for standardized documentation and markup of casework in order to do these comparisons across agencies, for example, is important. The extent of consensus on decisions, verification and blind verification, as well as proficiency testing are all implications that come out of this work. Just to highlight a few, I'm going to skip that, and we'll talk about lack of consensus on decisions. One of the issues is most of the agreements or most of the disagreements or change decisions that we saw related to inconclusive decisions versus erroneous individualizations or exclusions. So it really was in that inconclusive category that we had to look at. And one of the things you need to be concerned with is whether or not you're going to have a single examiner's decision stand. We know that a single examiner's decision will not necessarily be reproduced by another examiner, especially in those complex comparisons, and especially when we're talking about things like inconclusive versus identification or exclusion borderline decisions. So then there's questions of quality assurance and verification and blind verification that fall out of that. So blind verification should be effective at detecting the erroneous IDs, but most agencies don't implement blind verification. Blind verification would detect only the majority of erroneous exclusions, but blind verification is not typical in exclusion determinations, and you should not expect value decisions to be reproducible, and no value determinations are usually not verified, so that means that you lose evidence, potentially. Proficiency testing, there's a lot to be said about the multidimensionality of examiner skill. The examiner skill varies substantially between examiners, and error rates really are not the only measure of examiner skill. So this has implications for black box evaluations, only provide a discipline-wide assessment of examiner skill or examiner competency should really be tested with proficiency tests. And proficiency tests should be rigorous and perhaps in a black box style testing. Other black box and white box studies have been conducted and are being conducted, and we do concur with PCAST, who called for more than one study to reproduce or replicate, verify those results. Mr. Peter mentioned the other studies that are ongoing at the FBI, but there are other studies in black box studies going on at other agencies and that are supported by the US government as well, that are listed on the slide. And I follow up by breaking down the references for anyone who wants them. And for any additional information, you can contact myself at the email address listed, or my colleague Austin Hicklin at Nobles is there as well. So thank you for your attention, and I'll be happy to answer questions if we have time. At the end.
Good morning, everyone. I know what you're thinking of. Am I going to make it by lunch? I'll really try hard. We're just going to talk about some of the research we're doing at NIST. I'll have to add the fact that not only NIST is doing this research, but we have a lot of collaborators we work with on specific programs, and one is the Firearm and Toolmark research. This is going on worldwide, even though we're going to be presenting our work here, and it's going to be at about a 30,000 foot level because we're doing a lot. disclaimers are the usual ones. You might see some brand names. Doesn't mean anything other than identification. And my comments are my own. Just real quick, going over tool marks. I know this is a varied audience, both here and on internet. But when we talk about tool marks, this is what we're talking about in a forensic laboratory. Two basic types of tool marks. That is striated tool marks, and that's a highfalutin word for scratch marks. And then also there's impressed tool marks. Both of them are important to understand in that their approach to finding a quantitative measurement of agreement means that we have to look at both of these independently or differently. When we talk about tool mark identification, we include firearms. Firearms is the most common tool used in criminal investigations, and the tool mark evidence is found on the fired bullets and casings. Non-firearm tools are also important in investigations, and they're what you'd expect, the hand tools that might be used to break a lock, open up a safe, or pry open a window frame. A little bit of an autopsy of a modern cartridge. It's composed of the cartridge case, contains the powder, the propellant. Sits in the top of the case is the bullet. The true bullet is the projectile. And the primer is basically the spark plug. It gets everything going. It gets impacted by a firing pin or a hammer, and it causes a flame to be jetted into the powder, and that ignites the powder and drives the bullet down the barrel. So the components that we're most interested in as evidence is the fired bullet and the fired cartridge case. Regions of interest most commonly looked at in forensic work is looking at the land engraved and groove engraved areas of the fired bullet. And you can see here the areas on a fired bullet. This would be the land engraved area. This in between would be the groove engraved area. It's important to note that the number of lands and grooves in the twist that's involved in the barrel engravement is class characteristics. Much like in fingerprints, there's loops and whorls and things like that. Similarly, there's class characteristics, and much of the comparisons done initially in the firearms laboratory. Regions of interest on the cartridge case most often is at the base. They're on other places on the cartridge case, but what we're going to be looking at is what's called the breech face mark. That's the impression from the back of the chamber onto the soft metal of the primer. The firing pin impression. There might be some aperture shears, that striated tool mark, and then sometimes the ejector is important in the identification. So that gets you kind of oriented. Current practice, most crime laboratories, is they look at the class characteristics first between the samples or the test-fired suspect firearm and see whether they fit the general group, the caliber, the number of lands and grooves, the type of the shape of the firing pin impression, what kind of impression was made on the breech face. And is there agreement there? Once there's agreement, then you can start evaluating the more individualizing marks. The opinions might be on a scale, but most coarsely they're an identification in the most currently used parlance. Exclusion, inconclusive is that there's not enough agreement one way or the other. And unsuitable means that in real life, fired bullets don't necessarily give enough surface area to be compared, and it might not be suitable for any comparison. Another common use is looking at automated comparisons. These systems, not only in the United States, are worldwide, and the basic philosophy of these is you take your fired casings and bullets at crime scenes, image them into a database. Also, any seized firearms, the test fires of those are entered in the database, and associations might be produced by those comparisons looking into a database of different cases. So you might have an exhibit to exhibit comparison, casing to casing at two different crime scenes that you didn't know were linked. Or you might have a recovered firearm, test fire it, and see what kind of crimes it might be involved in, the firearm might have been used in. Now, to be clear, this is a sorting system. It's like a Google for firearm evidence. It makes all those comparisons and it scores them, but the score is helpful only in ranking. The score itself is not a measurement, a statistical measurement of the potential error in identification. You still need to recover that evidence. The examiner has to take the actual evidence and compare it under a comparison microscope. If you need more, I'm going to talk about a presentation or a publication that's free. If you go online, you can get a PDF of this. It's the most fundamental items that I just talked about. I helped get published through the National District Attorneys Association, and it's online. So if you want to get the basic information on what goes on in a crime laboratory and how this evidence is analyzed, please take a look. Obviously, why we are here is that much of the current practices, including firearm and tool mark examinations, are under scrutiny. We all know the report in 2009, the PCAST. Both of them are talking about the statistical or fundamental elemental foundations of the science, and that they need to be more empirical studies to just test the system and the examiners to see how well they make their conclusions. And also on the bottom, and I'm going to talk about this more deeply, is the comparisons are made by a subjective method. You might make a comparison side by side, but the determination of sufficiency for an identification, whether it's a firearm or fingerprints or whatever, it's subjective. Now, the subjectivity might be very accurate in the long run. We're going to test for that more. But wouldn't it be nice to have an objective mathematical method to look at those surfaces and actually independently measure how much agreement there is, and then relate that agreement to maybe a statistic? So, one thing we have to understand is tool marks and ballistic evidence, they're not like DNA. Earlier on, there are some comparable items between the two, but fundamentally they're not. In DNA, what we're looking at are class characteristics. That's where you look ahead of time for variability in certain areas on a gene, and you can measure those by looking at a lot of people and find out how these variables show up in the population. DNA profiles don't change. And you can always work on your statistics by looking at population samples. Tool marks, they're not known in advance. You cannot predict what the tool mark is going to look at until you actually test fire it or actually examine the comparison. They might change over time. There's different ways firearms get manufactured. So that adds another challenge. And your population data currently, and we're working on it, it can only be assessed through comparisons to tool marks when you know what the ground truth is. This leads into the key challenges we're working on as a science and primarily at NIST. There's significant variability in the marks observed from a same firearm. There can be differences in firing conditions. That is, you might use different brands of ammunition. They might have different pressures. The pressures we're dealing with in even modern handguns, we're talking about tens of thousands of pounds of square inch pressure. The bullet and the cartridge case actually flow and form to make these marks. So if you have variation in that, that really adds to the potential differences. Evidence itself can be deformed. Cartridge cases could get run over in a street. Bullets get fragmented. The use and abuse of the firearm might have as it's being used through the years. It's from the time it's been made in the factory versus the time it's recovered at a crime scene. We don't have any control of that. We can only do a lot of tests to see what kind of abuse might cause certain changes or just basic wear. Ammunition components, they themselves have tool marks made by the factory. So those must be included in your assessment as to the comparisons. And what kind of measurement methods you're going to be using. Is it going to be the simple comparison microscope, or are you going to be getting into the 3D, and that's what we'll be talking about. That's where it's going to. There's similarities on the marks, even though we know they're from different firearms. That can be caused by accidental marks caused during the manufacture of the firearm, being carried over between multiple firearms. Those firearms make evidence, and the coarse marks might be confused with an identification when you'd be wrong. Manufacturing effects also transferred of firearms evidence. There's no full consensus or objective, quantitative, for the comparison criteria. Not yet. We're working on that. And there's no consensus on to express the weight of evidence. In this case, it'd be how much uncertainty do you have in your conclusion. And there's no comprehensive ground truth population data yet of tool marks, but we're working on that, and we'll describe some of that work. We're definitely seeing a sea change in the crime laboratories when it comes to firearms evidence. It's going from the 2D visual comparisons to the 3D metrology. There's a lot of advantages in 3D capture of the surfaces of fired evidence. You can reproduce those surfaces. Many of those, almost all of them that I know of, are being measured in standard international units. You can measure it once, and you can move those images around amongst other examiners for peer review, for quality assurance, for training. And since they are measurements, now we can use those to actually use algorithms to see the similarity, and then also in the populations determine a confidence or a probability. The status, you're already going to be starting to see the 3D image comparisons, the virtual comparisons being used in casework. And the virtual microscopy is probably going to be the next stage from the comparison microscope to virtual comparisons. Before 2009, NIST was heavily involved in making standards for use in the NIBIN system, the ballistic imaging system in the United States, and tested amongst all the laboratories in interlaboratory testing. That came about with the development of the standard reference material bullet. They're basically a reproduction of the types of land impressions you'd find on actual fired bullets, and a cartridge case standard. Those are all near identical standards with their own surface profile that's part of the certification. Those were initially used for optical systems. The NIBIN program then at that time was mostly optical, but necessarily we needed to make sure the network was working correctly. However, in the development of the standard bullets and cartridge cases, NIST had to develop some kind of metrics that when these were being produced, how close were these being produced? Do they meet NIST strict quality assurance? And the algorithms and measurement processes used to qualify these standards were some of the basic algorithm methods used in later research in actual fired bullets and casings. So there's a pedigree of what we're doing going back to the standard bullets and cartridge cases. We're also making standards for the calibration of the new developing technology that's going to be the future 3D comparisons. You can't tell whether you have an accurate surface until you have that system calibrated. So what we're developing through NIJ funding is a suite or a group of standard surfaces that the systems can be used to, whether they're confocal or they're a metrological system. No matter what kind of method, they can be used to test against known standards. And that is, here you can see half spheres we're considering, grids. Mirrors are important because that's the flat that you have to start with. Chirp targets is an example of basically what kind of resolution your system is looking at, and roughness scales. Just some of the ideas were coming out. This is very close to being manufactured right now. It's being evaluated as to the design. We also work on documentary standards. You've seen that already. The ISO 17025 is one of the accreditation manuals that most crime labs work toward. ANAB and other, ASCLD Lab and other accreditation societies and groups independently accredit crime laboratories using these standards. They're also being used as developing new standards through the Academy Standards Board, ASTM, and the organization OSAC is also helping prepare those standards and evaluate those standards for later SDO registry. There's another group, the ASME and ISO group is general product specifications for surface texture measurements. There's already a standard group that's used in industry and also in members in NIST are part of, looking at the standards for surface metrology and how they adapted to firearm and tool mark examinations. So we have a lot of people involved in even the standards development. So most academic establishments have a hard time even getting a gun on campus. So how are they going to be able to look at these algorithms that might be useful in the future? They might have a great idea, but if they all they have is 20 bullets, how rigorous is that comparison? So one of the things we developed early on at the NIST is a ballistics toolmark research database. This is research samples only. In that there's multiple bullets and cartridge cases of specific tests. There's some that are like one single firearm fired many thousands of times, and samples of those are actually imaged. Everything from consecutively made breech faces, barrels, all those type of test samples are in here in 2D and almost all in 3D. So some of the statistics, again, this is NIJ supported. We have contributions for over 30 laboratories. We not only measure them, but we also get samples from crime laboratory collections. So it's a nice nationwide selection of types of samples that examiners are going to find all over the United States in their casework. So we have over I know this is a little old, 500 firearms, 6,000 measurements. The firearms collected, they're waiting to get measured, are over 5,000. For this toolmark database, there are 144 registered users, and just so you can see the usage statistics is going up as more and more academic establishments, also companies, industry who are interested in getting into this space. This is a great place to go to where you can get lots of different types of test samples and be able to test your systems. They're all in a form that is X3P, so the sample measurements are all interoperable. If you know if your system can compare X3P samples, you can use this data. So the objective quality measurements, there's two different ways of looking at it. Looking at specific individual marks, let's say this is a base of a cartridge case. And or you can look at the whole area of the impress. This is a false color of a 3D impression from two different cartridge cases. We also do it for the bullets. What are the features that are of comparable interest that start lining up, in this case like consecutive matching stria? Also, we can also look at these samples on the bullet, one-on-one, totally, one land impression. But the problem is when you look at the full sizes, casings and bullets don't mark equally from firing to firing to firing. So there's going to be a difference in these impressions. So if your algorithm looks at everything to everything, there might be an identification, but your score might get depressed. So we've done from that is break it up. And this is kind of what an examiner does under a microscope. What happens is the reference topography is broken up into cells, and each cell is independently marched around into that system, into what's going to be compared to see whether there's any agreement in 3D. And that's what's called congruent matching cells. There's a whole family of these that we're evaluating, and they're showing real promise. So here's an example of how it would work. Here's a surface of a cartridge case. Breech face is broken up into squares, and then it's compared against another cartridge case fired from the same. We know that this is the same firearm that caused this cartridge case. It's independent as to rotation. This is fully automated. The only thing that is not automated is there has to be a limitation as to cutting out the firing pin area. Here's an example of reference A back to a different cartridge case. You might have a lot of the same values, the same texture, but it just can't find any congruent matching. So what we're doing is we're taking our match scores and relating it to the number of congruent matching cells. In this case, there were no congruent matching cells, and here there's 24. For an example, for bullets, we're dealing with stria, and the stria has to be decomposed into a profile of the stria, and the profile is what gets analyzed. Under a 3D microscope, this is the type of the detail. This is a nice example for tours and presentations. It's one single land impression. So this profile along here is what actually gets compared. So one way is we break up the profile into segments, certain type segments, and we march the segment down from the referenced onto the compared, and we match, see what kind of match score happens. So when it gets into a locked position, you can see that spike. That is the fundamental part of how we do comparisons objectively. And as these segments get fit, another segment gets run, another segment gets run, and then you actually count how many segments are in agreement in the profile comparisons. I know we're going very high over the technology, but all this stuff is getting published. In fact, this profile segment analysis scheme has been published already in Forensic Science International. So now this is the nut, right? This is what we're going to talk about. We got enough agreement. We have a score. What does the score mean? Well, the way you can find it out is to actually look at populations of known matches and known non-matches. In the perfect world, we'd have a nice discrimination. Anything above a certain critical value, we could call it an identification. Anything below it is an exclusion. But we know the world doesn't work that way. And we're starting to see these type of distribution examples in more than firearms and tool marks, but also in shoe prints, fingerprints, things like that. So here's the same source comparison. We have a distribution of the scores. Scores on the bottom, probability densities on the side. Same source, different source. Your critical value might be the crisscross of the two distributions. The question might be, okay, here's my evidence score. What does that mean? Well, one way of looking at it is looking at the false positive, false negative rate. Over here in the blue would be a cumulative false positive rate. Over here would be a cumulative false negative rate. So we can look at these probabilities, make some assessments as to your system and things like that. But most importantly, we want to find out if we actually had casework, how are we going to be able to describe that evidence match? One way is to do the likelihood ratio. And a likelihood ratio in this case, using these two distributions, would be, here's your evidence score. The height of that would be the probability of comparison of the known non-match compared to the full probability comparison score the same, a true match. And you actually weigh the two into a ratio. Here's the beginning of our population studies. You can see these are ranked as to the type of firearm being used, and you can see pretty significant differences in the match scores. I bring your attention to the known non-match scores. You can see very similar distributions that fall right off. So since it's not DNA and we don't know what it is, you have to do empirical databasing to get an idea of what kind of distributions you're dealing with. We're working with FBI and also the Netherlands Forensic Institute to be able to give this type of product for the firearms examiner, where the comparison is made. You might have multiple vendors or different instruments. They all might have their own, may or may not have their own calculations as to the agreement. That score can go into what's called rapid fit, and the demographics of that comparison can be compared to the demographics of a relevant database. Here's how it might work. Here's your cartridge case of the crime scene, suspect firearm, we test fire it. The examiner does a comparison microscope work. Subjective opinion might be, yeah, that looks like an identification. Then it goes into 3D measurements, goes into correlation algorithm, and the score is, in this case, 23 cells, right? So the metadata is that's a Glock, cartridge case, parallel lines, broach, preface, nickel primer. There's where 23 cells would be, looking at a population of similar demographic. And that would be translated into a statistic. Alicia would help on that. You have 2 cartridge cases at a crime scene. You don't have a firearm yet. Do the comparison. You might have a database search through NIBIN or some other system. Firearms examiner actually qualifies if there's an identification. In this case, there goes through the same correlation algorithm, but what's different is that it's a different database. In this case, we don't know the firearm, but we do know it's a 9mm, it has a hemispherical firing pin, and it has a nickel primer. So all of those get compared, and what kind of distribution it might be, 20 cells, right? So that's how we are envisioning how this could be used in crime laboratories, where the scores that they get, their match scores they get, can be related back to a database that everybody's going to be sharing. So, database searches, common practice, we see about the news all the time. Virtual comparison microscopy, that's already been validated and it's already starting to be used in casework. Now, the objective identification and validation and statistics, that's still out. We're working very hard on that. So, let me say what the outlook is, the happy news. I think we've got a lot of significant research efforts done since 2009. And they're finding their way into the laboratories. 3D metrology and virtual comparison are just beginning to be used, and the computer-aided techniques will help augment but not replace the examiner. And here's the NIST contributions: improvement of the measure methods, quality control, research and databases, objective comparison methods, and estimation of the weight of evidence. This is a big team. We've grown over the years. We have a mixture of not only metrologists, but also sensor experts, statisticians, and I'm sitting in there as the examiner, SME. So it's looking up, I think. I'm here to say I'm very excited about where we're going to be in probably 5 or 10 years. Okay.